And we're going to be looking a lot at hemoglobin and heme. Okay, heme is a prosthetic group of hemoglobin, and heme carries oxygen. So here is heme, right here. We've got this central iron atom, which is going to carry oxygen, and then you have this structure of carbons. It's called a porphyrin ring, right here, porphyrin ring. Okay, here's an edge view, and so we would take this image and rotate it so that this edge is looking at you. Okay, heme has the central iron, and it binds to oxygen, but it also binds to histidine. So it binds to these two things in addition to the carbons and whatnot in the porphyrin ring over here. But of most importance is it binds to oxygen and histidine. Here's a graph. On the x-axis we have concentration of ligand, and then on the y-axis we have, I think that's theta, but that stands for percentage of ligand and percentage of binding sites that are full with ligand. So percentage of binding sites that have a ligand. So this would mean 50% of the binding sites have a ligand. We're going to introduce a new term, KD. This is going to be an important term for you guys to know. And so the KD is a concentration of ligand, okay, because it's on the x-axis. So KD is the concentration of ligand when half your binding sites are full. That's one way to define it. Probably the more important way to define it is written down up here, and if you have a smaller KD, that means you're going to have tighter binding or tighter affinity. Whereas if you have a larger KD, if you have a higher number, you're going to have less tight binding. You're going to have less affinity for your ligand. So let's look here at smaller KD. So if we had a smaller KD, let's say we shifted KD over here a little bit because we made it smaller, we would expect to have tighter binding, and we would have a lower concentration of ligand needed to reach 1/2 of the binding sites being occupied. But the most important concept to know is that if you have a small KD, you've got high affinity or tight binding. If you have large KD, you have low affinity or low binding. Alright, here's a table, and we have a couple protein names over here, and then we have some KD values over here. And so remember, KD tells you how tight this protein is going to bind the ligand. So which protein will bind its ligand more tightly? Avidin or calmodulin? And so remember, tighter binding is with the smaller number, so we would expect avidin to bind more tightly. Ooh, I should maximize this. Okay. Alright, so we've got protein X and protein Y. And we've got a concentration of ligand right here. So which protein, X or Y, has the greater affinity for ligand A, for this ligand? So the answer would be X. Protein X has a greater affinity for ligand A because it needs less ligand to have its binding sites half full. And so if you think of the binding site as a magnet that attracts the ligand, the magnet or the binding site of protein X is going to be stronger than the magnet in the binding site for Y. Or in other words, X has a greater affinity for its ligand than Y does. Okay, here's heme, and remember it binds oxygen and histidine. Okay, notice that when iron binds oxygen, it's bent. Okay, and here's carbon monoxide, and it's linear. And when things bind in a linear fashion, they're more stable. And it turns out that carbon monoxide binds to free heme more than 20,000 times better than oxygen. Or in other words, the KD for carbon monoxide is more than 20,000 times lower. So when I say free heme, that means that the heme is not in the hemoglobin. It's just free heme. And so this is why carbon monoxide is so poisonous to us and so dangerous. Because if carbon monoxide is taking up this binding site, then the heme cannot carry oxygen. And we essentially starve our body and our cells of oxygen. However, when heme is bound to myoglobin or hemoglobin, so when heme is not free heme, but when it's bound to hemoglobin, the carbon monoxide is not 20,000 times better, but it only binds 200 times better than oxygen. So why the difference? It has to do with structure. And so here's heme, and then it's inside of hemoglobin. And hemoglobin has amino acids that are strategically placed to make it very inconvenient for carbon monoxide to bind. And so this histidine residue right here is just in the way. Carbon monoxide really has a hard time being linear right here. It can't really fit that well. And so steric hindrance prevents carbon monoxide from binding. And so we see how important structure is to function. Okay, we're going to introduce a new concept about hemoglobin, and this slide is going to be really important. So after I've gone through it, maybe just pause it and just look over it a couple times, because you'll definitely be tested on some of these concepts. Hemoglobin has two conformations. It has a T-state and an R-state. Okay, so it turns out that the R-state binds oxygen better. And so as a mnemonic, to remember that the R state is when you have oxygen bound, just think roxygen. Okay, so you put an R in front of oxygen and you just say roxygen, and then that tells you what state oxygen is bound to. And so when you have oxygen, it's going to be the R state. There should be a little G in there, but you get the idea. So the R state favors oxygen binding. And so when you have this T-state conformation, you have no oxygen present. And then also, when you have the T-state, the shape of the heme is puckered. And so a mnemonic for that is tuckered, puckered. And so the tuckered is just to help you see the T right there. T means that it's puckered. There's something called the TR transition. And so remember, this is really a heme molecule that I drew right here. And it's in the T state, so it's puckered. And so that means it's shaped kind of like this. It's puckered a little bit. So we have the T state. We have no oxygen present. What's going to happen is oxygen is going to bind to the T state, and the conformation will change to the R state. So we have the R state. We have oxygen. We're no longer puckered. We're planar. And so usually when we have a conformational change, there's breaking or making of weak bonds. And so when oxygen binds here, it causes breaking of some bonds to change conformation. And in biochem class, you'll probably hear those bonds referred to as salt bridges, or like a bunch of ionic bonds. Okay? So here's the T state right here, and I just circled this to show some of the salt bridges, some of the ionic bonds. Remember, when you have the T state, we have no oxygen and we're puckered. Here's another picture of the T-state, and this is just showing the salt bridges. And so all these dashed lines represent these ionic bonds. So we've got positive to negative charges everywhere. So oxygen would come in here and it would disrupt some of these salt bridges, and it would cause the conformation to change from T-state to R-state. Okay, so here's another picture. Here's a picture showing the TR transition. So here's the T-state, and we would have some ionic bonding, some salt bridges going on, but then oxygen would bind and cause the R-state conformation. And remember, the mnemonic for that is oxygen. Here's a picture showing the puckered and the planar state. And so this is supposed to show that it's puckered, and the T-state, so the T-state is puckered. So remember, that's puckered. And then the R-state is, you can see it's a little bit more planar, and you've got the oxygen bound there. Alright, so when you show this on a graph, hemoglobin is shown as what's called a sigmoid curve right here. And so these two curves, this top one right here, this really steep curve, and then this more shallow curve, these are kind of hypothetical curves. Okay. So we have a problem, because if hemoglobin binds oxygen with high affinity in the lungs, it would efficiently pick up oxygen in the lungs, which is good. You know, that's what we want. However, because the affinity is so high, it would never, the hemoglobin would never release the oxygen to the tissues. And so in this state, if we had a really, really high affinity state, it would hold on to that oxygen too tight in the tissues and in the lungs. So the body would never get its oxygen. And so the solution for that is the T and the R states. And so we know that the R state has a higher oxygen affinity, and so in the lungs we would have a higher oxygen affinity. We'd see the R state, and then in the tissues we would want a lower affinity so that we could drop that oxygen off, and so we'd see a T state favored. And the way we favor T and R states is by something called the Bohr effect, and we'll see that later on, and that's affected by pH. Okay, there's a term called cooperativity. So hemoglobin has cooperativity of oxygen binding, which means once a single oxygen has struggled to bind, and so initially that binding of O2 is difficult, but once that O2 has bound, then each successive binding of oxygen becomes easier. This picture's kind of crazy, but I don't think it's that helpful. But you've got this snake-looking thing, which represents some of the different subunits on hemoglobin, and you've got this binding site that's going to accept oxygen, and this pink thing represents that the conformation is not very tight, and so oxygen comes in here and it just kind of has to struggle to bind, but then it causes this next oxygen to bind more tightly right here. Okay, this is called the Bohr effect, and so remember, we don't want our affinity for oxygen to be super high in both the lungs and the tissues. You know, we don't want to have the R state in both the lungs and the tissues, and so we can make sure that that doesn't happen by this thing called the Bohr effect, this idea called the Bohr effect. And the Bohr effect tells you about the effect of pH and CO2 on oxygen binding. So we have three sigmoidal curves here. And so this middle one is a pH of 7.4. This curve on the left is a pH of 7.6, and this represents the lungs. And then this line right here, pH of 7.2, represents the tissues. So if we have a higher pH, we're going to be in the lungs, and if our pH is higher, then that means our hydrogen concentration is lower. It means our CO2 is low, and think about our concentration of oxygen in the lungs. We've just breathed in air. Concentration of oxygen is really high in the lungs, and so we want the affinity of hemoglobin for oxygen to be high. So in the lungs we see our O2 affinity increase because of the low hydrogen and CO2 concentrations. And so we see this graph shift to the left. If we're in the tissues, we want to dump our oxygen, so we want our affinity to decrease. And so this should say higher. Okay, higher. We're going to have higher hydrogen concentration. We're going to have higher CO2 concentration. If we have high hydrogen, it makes our pH decrease. Okay, BPG, also known as 2,3-bisphosphoglycerate. Look how negative this molecule is. It's really, really negative. Okay, and BPG is going to regulate oxygen binding to hemoglobin. So if BPG is present on hemoglobin, it decreases the affinity of hemoglobin for oxygen. And so there's an inverse relationship. The more BPG we have on the hemoglobin, or in the red blood cell, so the more BPG we have, the lower the affinity for oxygen. Alright, so BPG would stabilize which conformation of hemoglobin, T or R? And so remember, BPG hates oxygen. So think about it like that. BPG, if it's present, you're not going to bind oxygen. So would it be roxygen or T? So it would stabilize the T state. How does it stabilize the T state? Remember, BPG is really negative, so it helps keep those salt bridges together. And so if we think back to the TR transition, when we're in the T state, let me flip back. Okay, when we're in the T state, we've got a bunch of these salt bridges. And so oxygen comes in here and disrupts all of these salt bridges to favor the R state. But let's throw BPG in here, really negative molecule, and he's just going to reform a lot of those salt bridges, which will just pop oxygen out, and oxygen won't want to bind, and we're going to have the T state favored. So let's go back. Okay. Okay, so we have a problem when we're at high altitudes. At higher altitudes, you have less oxygen, and so less oxygen gets delivered to the tissues, and so that's a problem. The solution is to increase the concentration of BPG in your red blood cells. And so how would that help? If you increase BPG, you decrease the affinity of hemoglobin for oxygen, so that the hemoglobin does not want to hold on to the oxygen for very long. And so it turns out that that BPG doesn't affect the lung's ability to pick up oxygen, but it does really have an effect on how quickly hemoglobin will dump the oxygen. So because the affinity is low, once the hemoglobin is delivered to the tissues, it's going to unload the oxygen very quickly. And so it results in quicker dumping of oxygen to the tissues. And so the transport of hemoglobin from lungs to transport of oxygen from lungs to tissues is much quicker, and it results in your body getting the oxygen that it needs. Okay, here's sickle cell anemia right here. This is a picture of it. That's a disease of the hemoglobin, and it results in a single amino acid substitution of val instead of glue, and that results in sticky spots. And so what we'll see is interaction between hemoglobin molecules. They'll congregate together and they'll aggregate, and they'll form these strands, and these strands will then aggregate together to form fibers. And the problem is this is all happening in a red blood cell, and this just gets bigger and bigger and bigger, and eventually the red blood cell will burst, and that results in anemia, weakness, and shortness of breath. That's the end.